Hello students, today we will start discussing about the new module, that is structure of biopolymers. In the very first segment we will talk about some basic biopolymers which dictate heredity and perform several different tasks inside any living organism, and those are DNA, RNA, amino acids, proteins. So before we begin, let us just first understand the importance of these biopolymers. So, any living matter that you see around us, you can relate them directly. If you want to relate life to a specific molecule, then that particular molecule will be DNA. So, living matters are in general composed of mainly carbon, nitrogen, hydrogen, oxygen, and some sulfur and phosphorus, right? So carbon is the most abundant element in life and is found in the majority of compounds that are present in living organisms. So most of the polymers that we were learning so far, that is why the main chain of the polymer spine, we considered their carbon to be elementary there, right? So organic compounds such as nucleotides, amino acids, and monosaccharides, they act as monomer units for much more complex biological molecules like nucleic acids, like DNA, RNA, proteins, and polysaccharides. So these eventually form a large set of biopolymers which further organize and assemble into cells, tissues, organs, and finally a whole organism. So let us first talk about nucleic acids. In nucleic acids, the first and foremost that we need to consider is obviously DNA. So in terms of understanding nucleic acids, the first thing that we need to focus on is that nucleic acid serves as repositories, or you can say transmitters for genetic information. So they are polymers of something which are called nucleotides, and we will come to the structure of nucleotides, and they are held by a phosphate bridge, right? So there are, we will come in detail of the structure. So there are in general two types of nucleic acids, namely deoxyribonucleic acid, deoxyribonucleic acid, that is DNA, or ribonucleic acid, that is RNA. So these are the two types. So DNA is organized into genes, which is basically found inside any living cell. You will find generally a portion which is called nucleus, and inside that nucleus you generally find these DNAs, and they are the fundamental units of genetic information. So sometimes we also refer to them part of DNAs which are having some important information. We call them genes also. So if we look at the structure of DNA, there are certain things that we know some information in bits and pieces, like they are polynucleotide chains, name of some certain names are associated with it, like adenine, guanine, cytosine, thymine, these names, and phosphate groups and so on. So we will look into the further details of these four kind of bases, that is A, T, C, and G, their detailed structure, and the detailed structure of DNA as well, right? So the most important thing that I think everyone remembers about DNA is the double-stranded helical structure. We will also talk about that structure. So you know that James Watson and Francis Crick worked on this three-dimensional structure of DNA based on certain findings by Rosalind Franklin. So the men got Nobel Prize for their invention, and if you look at this double helical structure, that is the key structure that everyone understands about DNA, is this ribbon model, where you see two ribbons, two ribbon like strands organized in a helical structure, where you see these step like connections in between, which are basically formed by hydrogen bonds. So let us understand this structure in much more detail in today's lecture. So the first and foremost thing that we need to understand are the basic part of the DNA or RNA is basically a sugar. So the RNA contains D-ribose. This is the structure of D-ribose, and DNA contains deoxyribose, right? So these are the two sugars. They differ in structure only at one position, where you will find this OH here, and here you will find only H. So that is the only difference between D-ribose and D-deoxyribose. So this is the one oxygen less than ribose, right? So this is the structural difference. So these sugar molecules are basically the fundamental units that hold the two strands together. So after these sugars you have bases. So there are 4 nitrogen bases which are characterized into 2 groups, are called purines and pyrimidines. So purine, as you can see here, purine and pyrimidine. So these are the 2 groups that they are purine and pyrimidine derivatives that are found inside nucleotides. So they are basically aromatic heterocyclic compounds. Purines are numbered in anti-clockwise direction, while pyrimidines are numbered in clockwise direction. So in DNA and RNA you contain the purines are the same, that is adenine and guanine. So these are the amine bases, right? So what are ATGC? They are amine bases. So A and G, that is adenine and guanine, the purine family, they are same in both RNA as well as in DNA. On the other hand, in the pyrimidine family, cytosine is same in both DNA and RNA, but in DNA you find thymine, while in RNA you find something called uracil. We will come to that later. So this is more or less the basic structure of the strands. So what you see here in the basic structure, that we have 3 parts. We have sugar, we have phosphate groups, and we have these amine bases, right? So how are they connected? They are connected in this fashion. So this entire structure together is called a nucleotide. So you have the phosphate base connected on one side, while on the other side you have the amine bases, and in between you have the sugar sitting there, right? So this is more or less the structure of the nucleotides. So if you now look for some basic characteristics of DNA, let us just summarize it, that it's a chemical substance or chemical molecule which is basically a biopolymer, and which is present in all living cells, right? And they control the chemical change in each and every cell, and they try to dictate that how the dynamics would go if we are looking at a living cell. They also decide, DNA also decides, because from DNA you will see later on that from DNA it is decided that which proteins are going to get produced inside the cell and which proteins are not going to get produced inside the cell, and thus that basically controls the fact that these particular cell, what kind of cell it is going to be. Is it going to be a muscle cell? Is it going to be a nerve cell? Is it going to be a blood cell? That is also decided by this DNA, right? And obviously different kind of organisms, they have different lengths and different sizes of DNA, and we saw that they are made of subunits which are called nucleotide. Each nucleotide is made of a sugar called deoxyribose, a phosphate group, that is a bond PO4, and an organic amine base. Okay, so up to this part I guess is clear. So if you now look into details of this structure, so in general the sugars, they are represented by this pentagon like structure, and the organic bases that we see, the amine bases, are in general dictated by certain kind of ribbon like structure with different heads, so that they just bind. So you can see that thymine and adenine, they have structures which are counter intuitive, that they will match, they will set in one another. So the bond, the hydrogen bond would form, so signifying that thymine would bond with adenine and cytosine would bond with guanine. So that is the kind of structures that usually they are referred to. So if you look at a nucleotide and if you don't want to look at the detailed structure, you are looking towards something which has a PO4 and a 5 pentagon like deoxyribose sugar, and in one hand of that, one of the amine bases are attached, like adenine is attached in this particular nucleotide. Now how that forms a chain? The chain is formed like this. So you have one PO4, one sugar and one amine base. From this particular point, so here you have the PO4. From this particular point, another PO4 is bonded, and with that is connected the next nucleotide. So you see that the nucleotides get joined side by side like a long chain, forming a polymer. So the monomers just differ because the PO4 part is same, the sugar part is same. So the only difference in each and every monomer is that what kind of amine base is attached to that, what kind of amine base is attached to that particular nucleotide. So there are 4 options. So let us just now understand that some nomenclature. So the bases would be called adenine, guanine, cytosine and thymine. Nucleotides are going to be called, so let us just read it carefully, deoxyadenosine 5' monophosphate, right? Why 5'? Because it is attached to the 5th 5' head of it, right? So deoxyadenosine 5' monophosphate. It is the name of the nucleotide. And same way, the other nucleotides would be called deoxyguanosine 5' monophosphate, deoxycytidine 5' monophosphate, and so on. And these are the abbreviation DAMP, DGMP, DCMP and DTMP. So these are the abbreviations for the nucleotides. So you understand that what are the monomers, right? But how does this double helical structure is formed? So one side is just a chain where PO4 holds the two side by side sugars and form one chain. Another similar chain is formed on the other side, but it's because the sugar has rotated. You can see that this sugar has its head on this side, but this sugar has its heads downwards. So you can consider that one strand is just the opposite way oriented from the other strand. So they have just the opposite orientation. So we call that one side it's a 3' side, one side is a 5' side, and with that, otherwise you will not get the bases side by side, because if you orient them in the same fashion, then the bases will be in the other way around, right? This way the base will be elongated this way and they will not be able to bond. So you need to rotate the chain, you need to flip the chain so that the bases face each other, and once they face each other, their bonds, the hydrogen bonds form. So between adenine and guanine, there are 2 hydrogen bonds that are probable to be formed, and while between cytosine and thymine, there are 3 hydrogen bonds which can form. So it cannot be swapped. So adenine cannot have a bond with cytosine, or guanine cannot have a bond with thymine, right? It has to be A bonding T and G bonding C. That is how these 2 strands should be made, so that they are exactly complementary in nature. And then only this double-sided, that you have PO4 on both, PO4 backbones on both sides, and in between you have these amine bases bonding with each other, you can form this structure. So adenine forms a bond only with thymine and cytosine forms a bond only with guanine. Don't forget that. So from there we arrive at a rule which is sometimes also called Chargaff's rule. That Chargaff's rule says that DNA has equal number of adenine and thymine residues and equal number of guanine and cytosine residues, right? This is called Chargaff's rule. It's basically comes from the molar equivalence between purine and pyrimidines. So the double helical structure of a DNA is completely based on this Chargaff's rule, because otherwise this structure cannot be made. Okay, so there are 2 different bonds which control how the DNA should look like. The first bond is this hydrogen bond between the 2 bases, and the second bond is the bond on the side, that is basically a covalent bond, and that is happening by the PO4. So these are the two bonds that hold the structure together. So I guess you understand how this structure looks like. So phosphate group, sugar, amine bases, and when they combine, that basically looks like something like this. So this is the total structure if you want to visualize it along with the original molecule. So you see PO4, PO4, PO4 on this side, right? Let us just decode it from the simple schematics that we were following till this point. So PO4, PO4 from this side, and these are your sugars. Along with sugars you have adenine or thymine or whatever is bonded here. On the other side you need the thymine inside so that it can face the adenine, right? So for that, the head where you had the oxygen, that goes down here. So oxygens are below in this side strand, right? So this upper end is called the 3' end. How do you name this? So this is your sugar, and you name them in clockwise direction. So this is 1' position, 2', 3', 4', 5'. So if this side is up, that is the oxygen is on the upside, that is going to be your 5' side. On the other hand, if the oxygen is below, then you flip this particular thing. So let us just flip this in particular thing. If we flip it, the oxygen goes below, and what we have here is that, let us flip it and see. If you flip it, then the 3' side will go up, right? So if you flip it, the 3' side goes up. As soon as the oxygen comes below, down below, the 3' side will be up, right? So following that, you can see that this must be the 3' side, and that is why when you orient them side by side, this is your 3' end and this is your 5' end. So I think you understand that how the two strands of DNA are complementary, and how the two ends of the DNA, one side you have 3' end, one side you have 5' end, right? Great. So let us look at some basic pairing up. We understood that this is how the basic pairing up happens, and now the only thing left to understand is why this ribbon like structure, that you have PO4 on the both side, and inside you had this bases, why it cannot remain as like a ribbon like this. So it could have remained like a ribbon, right? Why it's not a tape like structure? Why do you have a instead of having just a ladder, you have a helical structure? So to understand this helical structure, you need to understand something which is called hydrophobic behavior, right? So the secret of DNA's helical structure could be basically because of the hydrophobic interior, that inside where you have amine bases, that part does not like water around it. So if that part is hydrophobic, then DNA would try to extract all the, because the nitrogen bases are hydrophobic, so it will try to extract or push away any surrounding water from inside, that is close to the ATGC molecules. In this portion, if any water molecule slips in, that is not something these bases would like. They are hydrophobic in nature. Phobic means phobia. It comes from fear, right? So hydrophobic means something which is afraid of water. So these nitrogen bases that we see here, they are afraid of water. They don't like the water in their surroundings. So DNA would like to push away the surrounding water from the nearby places of the nitrogen bases. On the other hand, the outside is much more hydrophilic, so they can have a large water exposure. So as a result, just like we squeeze after washing a cloth, as we squeeze to keep the water out from the inside of the cloth, if DNA has, instead of being a tape-like structure like this, if it rotates and makes a helical structure like this, then the probability of water molecules squeezing inside these amine bases, close to these bases, becomes really less, and thus it becomes easier for DNA to maintain its hydrophobic interior. So this could be one of the reasons, though it's still under research, this could be one of the reasons that why DNA is pushing away, why DNA has a helical structure. So let us just quickly have a look at some salient features of DNA double helix. The first thing is that it's a right-handed double helix. That means if you have, screws, right? How a screw could be right-handed or left-handed, how you rotate it depending on that. So depending on that, this DNA is a right-handed double helix. And what we call is that it consists of two polydeoxyribonucleotide chains, right? Poly, that means polymer of what? Nucleotides. What kind of nucleotides? Deoxyribonucleotides, right? So that's the name. It looks long, but if you break it by part, you will understand what it means. So polydeoxyribonucleotide chains twisted around each other. So each of these chains are complementary to each other in terms of base pairing. The two strands are anti-parallel. Just I said that one runs from 3' to 5' direction, other runs from 5' to 3' direction. Okay. And the width of the DNA, what is the size of the DNA we are talking about? So width of the DNA is around 2 nanometers. Each turn, that means each helix pitch, screw pitch, is 3.4 nanometers, around 10 base pairs, right? So 10 nucleotide pairs constitute of one pitch, one rotation. That means if you look into this picture, so from here to here, right, one screw pitch is here to here. So that basically usually contains 10 base pairings. Now each strand has a hydrophilic, that means one that loves water, hydrophilic phosphate backbone. That could be the periphery, that constitute the periphery of the molecules, the outside of the molecules, while the hydrophobic bases are packed inside the core, and that's why maintaining the helical structure, right? The two strands are held together by hydrogen bonds, where AT pair has 2 hydrogen bonds, GC pair has 3 hydrogen bonds. Thus GC pair is obviously 50% stronger than the AT pair bondings. Hydrogen bond formed only between a purine and a pyrimidine, right? So the pairing is fixed and the Chargaff rule exists. So that is about some basic features related to DNA, and some information about the double helix. So these are all numbers. You can hear some quantities like kilo base pairs or mega base pairs. They mean the same as they mean in usual physics. Yeah, sometimes base pairs are also mentioned by BP. BP means base pairs. KBP means kilo base pairs. That means 1000 base pairs. Mega is 10 to the power 6 and so on, right? So this is how the names happen. The very interesting thing that you need that you can understand from the size of the cell, as I said, the DNA is very thin, right? But it's very long also. So a DNA could be 1 meter to 2 meters long, and it is packed inside the cell nucleus, which is around 10 micrometer of diameter. So that needs a huge amount of packing. So that is another thing to understand, that how the DNA, long DNA, is like 2 meter long a chain can be packed inside a small cell, not only the cell but the nucleus of the cell. So this chart more or less gives you an idea that which genome, which particular species has how long or how small DNAs, and you will see that it does not, the size of the genome, that how long the DNA is, the size of the genome has nothing to do with how the species is more and more developed species. So you will see the flowering plants could have a very large amount of genome, that is the length could be very large. Insects can have very long genomes and so on. So it is completely, it's not very species wise constantly growing, or you cannot see a direct correlation with that. Now we will finish up the discussion about DNA by talking about something called replication. So replication is that before a cell divides, the DNA strand unwinds and separates, right? And each strand makes a new partner strand by collecting nucleotides from around, so that the genome can get doubled, and when the cell divides, the two cells get equal amount of DNA. So what happens is they separate, the two strands separate, the hydrogen bonds between them, that breaks and they separate. Once they separate, after that what happens is each strand start templating, start collecting base pairs from around. So around the cellular environment around the inside the nucleus, there are several such nucleotides scattered around. So what they do is they start making a new polymer in complementary way. So if you have a C, it asks for a G. If you have a C, it asks for a G. If you have a T, it asks for a A. And thus a new polymer chain starts getting formed. And then from one DNA, you start making two DNAs. And this is the secret of life. This is how replication happens. And this is how from one DNA strand, two DNA strands are made and a cell divides. A cell divides into two parts and having equal genome in both. So we'll stop this lecture here, and in the next lecture we will talk about RNAs as well as genetic code.